Hello.
Yeah, we're on camera.
How do you know?
I can see it on the computer over there.
Okay. Hi. Hello, everyone. My name is Sandra Splinter Bondurant, and I'd like to welcome you to this month's Biotech Talk that's being hosted today by the UWBC Gene Expression Center and BioLegend. I also would like to let you know that in May, we'll also be hosting another Biotech Talk, which will actually have two talks that are happening on May 16th. So it's a different date than our normally third Thursday of the month. So put it on your calendar, May 16th, we'll be having two more talks for the Biotech Talk. And those will be from Thermo Fisher and Dustin Rubenstein. In addition, on May 4th, the Gene Expression Center will be hosting a 10X Genomics webinar on May 4th from 10 a.m. to 11:30 a.m. This will be a webinar-based only talk, and there is information on the Gene Expression Center webpage for information on how you can log in or sign up to attend that webinar. So for today's talk with BioLegend, which BioLegend is giving, today Leesa Pennell is joining me, who is a field application specialist for BioLegend. Today, she's going to talk about the Total-seq products that are in the BioLegend portfolio that scientists can leverage in their single-cell RNA experiments that they're doing, whether here on campus or at another facility. So without further ado, I'd like to welcome Leesa to give her talk. And also, if you could hold your questions until the end of the talk, Leesa will address them at that time. Thank you.
Thank you, everybody, for attending. Thank you for the nice introduction, Sandra. All right. So everyone, laser pointer check. Hopefully this is all working. Okay. I'll knock it over. Okey-dokey. All right. So as Sandra mentioned, I'm going to talk today about our Total-seq reagents, which is an integrated end-to-end solution for single-cell multiomic analysis from BioLegend. So first, I'm just going to briefly, give a little bit of a background on BioLegend. A lot of people may know BioLegend for our antibodies for flow cytometry. And at BioLegend, our portfolios are broken down into 6 basic categories. Our cell analysis, which includes our antibodies for flow cytometry, proteogenomics, which we'll talk about today, molecular and cellular biology, so antibodies for Western blotting and other non-flow applications, our immunoassays, so ELISAs and Legendplex kits, clinical and diagnostic reagents, and then our custom solutions, which we can provide on an as-needed basis. So as I mentioned today, we're going to talk about mainly our proteogenomic solution, which is to address our antibodies that are conjugated to oligonucleotides. So for today, I'll go over some main categories here. So integrating Total-seq antibodies with your single-cell RNA-seq. So that application is known as site-seq. We'll also talk about cell hashing and our pre-titrated and lyophilized antibody cocktails, which give you 120-plex and beyond. And then if we have some time, there will be some little bit on other applications and our multiomics analysis software as well. All right, so diving right in terms of the potential of single-cell RNA-seq. So the current state of the art with single-cell RNA is allowing deep interrogation of cellular mRNA. So this is massively parallel. We can look at tens of thousands of cells at the same time on a single cell level, and we can look at thousands of different genes. And so, in this plot over here, we're looking at clustering of a different heterogeneous population into different types of cells based on their RNA profile. And then, there are different approaches that companies like 10X allow you to do, which would be 3 or 5' directional sequencing and VDJ sequencing for immune profiling, among others, but these are the ones that BioLegend products are compatible with at the moment. So in terms of protein analysis technologies, the current state of the art is to use familiar technologies such as flow cytometry, or colorimetric discrimination. So these are widely utilized and trusted. You can stratify different cell populations based on surface expression of different molecules. You can have readouts by flow cytometry or CyTOF. However, these are inherently limited by the number of conjugates that are available. And you get no or little simultaneous RNA data. So in terms of flow cytometry capabilities, with conventional flow with something like an A5 Symphony, you're looking at about 28 parameters. Using full spectrum flow cytometers, such as the 5-laser Cytec Aurora that's in the flow core over here, you can get around 40 plus maybe parameters. In CyTOF, you can do 40 plus parameters, depending again on available conjugates. So those are just some examples here. But then you're stuck. How can you add any more in terms of surface markers? So that's where we enter this technique called CytSeq, which is the cellular indexing of the transcriptome and epitopes by sequencing. So this technique was coined CytSeq by the New York Genome Center, which used antibodies coupled with oligonucleotides in a method termed CytSeq, which combined highly multiplexed protein marker detection with transcriptome profiling for thousands of single cells. And so in this case, the first publication, there were biotinylated antibodies that came in with a streptavidin with the oligo. And BioLegend took that a step further and made these antibodies in our TotalSeq portfolio, where we have antibodies that are conjugated directly to an oligonucleotide. That oligonucleotide contains a PCR handle for amplification, an antibody barcode that's unique to that clone, and then a capture sequence to make it compatible with various single cellularization technologies. And so because this antibody barcode is 15 base pairs, you can get over 1 billion unique barcodes, which means basically the clones are endless in terms of how many you could possibly add to your sample. And so, again, this is just that publication. It's been around for a number of years now, so people have definitely expanded upon that. So as I mentioned, we're merging protein analysis and single cell RNA-seq by using TotalSeq. And so we stain our cells in a similar fashion to flow cytometry before we go ahead doing our single cell RNA-seq. And then we have these antibody oligo tags, and they are captured in a similar mechanism to that of the mRNA. And then we construct an independent antibody-derived tag library. So basically here you've got your cells, you label them with the antibodies have the oligos, and those are going to get read by sequencing in the end. So in terms of the workflow, the antibody oligo tags, again, are captured similarly to the mRNA. We're going to construct this independent library. So what happens here is we have our antibody binding with the cells. Okay, so you can see these antibodies with the oligo tags. We're going to wash those cells very well to make sure that we remove any unbound antibodies. So that's what this guy is over here floating around. We want to make sure that doesn't get encapsulated in this droplet, because any unbound antibody will eventually get transcribed. We won't have any way to discriminate whether it was actually bound to the cell or not, because after this encapsulation, the cell lyses, and then the antibody oligos are released, as are the mRNAs. So at that point, the mRNA and the oligos from the antibodies hybridize onto the bead. In this case, this is working with something like the 10x genomic system. We have emulsion breakage, reverse transcription, and template switch in bulk. And then the last step here would be size selected cDNA for further library prep. And then we have a second size selection for the antibody derived tags. So some key takeaways here. Again, I'm going to stop saying the first one there and the second one, since I've said them a bunch of times, but basically their antibody derived tag and the cDNA library share the same cell barcode, so we can identify which cell it came from. And then each oligo in there has a unique molecular identifier, either the ADT or cDNA, so that we can quantitate how much of those are present within the library. So again, we have 10x gel bead over here. There's the oligos that are attached to the gel bead, and then we have our antibody, where there's a complementary sequence on the antibody oligo and the gel bead oligo to get those to bind to one another. So in the initial findings of doing the CytSeq technique, it was a 11 antibody immunophenotyping proof of principle experiment that was done. And so what they found with this was that the human ADTs were highly correlated with the cognate mRNA. So over here, again, we have clustering of these cells based on the RNA transcripts. And then what we have in this is the same cluster, but then overlaid with either the mRNA in blue or the ADT expression in green for these markers, CD19, CD11C, and CD34. And so what we can see here is that ADT detection can overcome instances of RNA dropout. So if you look at the CD11C mRNA profile, you see kind of a sparse expression on this cluster, which contains the CD11C positive, CD14s like monocytes, maybe some dendritic cells in here. And when you see the ADT overlay on top of this group cells, you can see that's really strongly correlated. So in this case, we know we would have probably lower mRNA expression of CD11C, but the protein is high. And by combining these two techniques, you're able to really further characterize your cell types in a population. There's a square to tell you what I just told you. Okay, so in the initial validation of TotalSeq as a technique, so what we did here is we looked at CiteSeq data and how it performs compared to flow cytometry and CyTOF. And so what we have on this side here is in a flow experiment, we labeled cells with a CD3 antibody and a CD19 antibody, and we're looking at those in bivariate. We can group here our T cells, which is about 52% of this sample, and then our B cells, about 13% of the sample. And then in a CiteSeq experiment, again, same clones we used, but instead of floors, they had oligos conjugated to them. And then we transformed the data from the sequencing output into these sort of this display where it looks a lot like a flow cytometry plot. And then we can see that you have, again, 52% of these are CD3 positive and 11% are CD19 positive. So very similarly matched across here. And then in the CyTOF experiment, using CD3 and CD4, and then in CiteSeq, same clones, but again, oligos instead of metals, again, CD3 by CD4, and you're getting similar proportions of these cells. So they're highly correlative, suggesting that, you know, we can really compare CiteSeq data to some of those standard technologies, the state-of-the-art technologies we already know and use. All right, so the last, no, not last thing. Another thing to mention here is looking at the RNA and protein differences. So we know that RNA and protein expression, they're regulated by different cellular processes. And so high RNA level does not necessarily mean high protein level, and vice versa. So in this case, what we have here, and this is the main plot I want to focus on, where we're looking at cells that are positive for protein but negative for gene, and then cells that are positive for protein and positive for gene. And so what you can see here in the ones that are really gray is something like CD57. So you have a lot of CD57 protein expression, but barely any mRNA at all. And so that'd be really hard to identify, similar to the example that I showed with the CD11C ADT versus mRNA. And so this was just done for a number of different proteins that we had antibodies targeting. And so some of them correlated 1-to-1, some of them, you know, you had positive for gene and protein, and others, you know, had a lot less gene expression, but still had strong protein expression. So this just, you know, shows that we're really able to get around the idea of RNA dropout by using this technique. Finally, something that was definitely brought up as we started, you know, showcasing this technology was, well, if I put all these antibodies on here, am I going to drown out the signal of the mRNA that gets captured by these gel beads? Am I going to be able to get any RNA expression at all? And so what we did here is we're using TotalSeq A antibodies. They share a capture sequence with the mRNA, so it's a poly-A tail on the antibodies. And so what we did here was we had antibodies added, more and more antibodies in the blue. So you can see those lines going up here. And you're able to still see that the median gene UMI count is consistent across the board, even when we're adding more and more antibodies to it. And then this was same for the median gene UMI and the unique gene counts as well. And so when we did overall antibody counts here, in this case, there was no antibodies added, and then this one had antibodies added, and you can see the overall gene counts, again, are very similar between the two. So some key takeaways from the validation that we did for this technology: antibody-derived tags are specific to those antibody targets. Those ADTs correlate with the mRNA. CiteSeq shows similar performance to both flow and CyTOF. It reveals differential patterns between RNA expression and protein expression, and the presence of these TotalSeq A antibodies, which again have the similar capture sequence to mRNA, does not impact the ability to detect genes, so gene detection. Okay, so this solution here can pipe into existing single cell sequencing protocols so that you can characterize again simultaneously the protein and RNA. We have a standardized barcode assignment of those antibody specificities. So I'm about to tell you that there is a number of different formats for TotalSeq antibodies, and the clone the barcode that we assign to a clone, let's say OKT3 for CD3, is the same across all formats. So once we know what that barcode is, we don't need to worry about trying to figure out differences between the different formats. We have a similar staining protocol when compared to staining cells for flow cytometry. So if you're familiar with flow, going to use this workflow is quite seamless and can be combined actually with flow cytometry. So if you do need to sort cells prior, you can do your labeling with the oligo-conjugated antibodies at the same time as your fluor-conjugated antibodies, minimizing handling of your cells so that you can preserve the viability. We know that those antibody oligo tags are captured in a method similar to that of mRNA, and we have this independent library. Great. So the formats I mentioned. So there are 4 different formats, 3 different formats for single cell RNA-seq, and then we have total-seq-D for single cell DNA-seq. So I'm not going to talk about that one today. And really, I would like to point you mostly in the direction of total-seq-B and total-seq-C, as these products are compatible with 10x Genomics workflows and will fully pipe into anything here that the GEC would use for their assays when working with your samples. And so in our total-seq-B portfolio, we have over 400 different catalog products in terms of single vial antibodies. These are compatible with 10x Genomics single cell V3 reagent kits for a dual index. There's a non-poly-A capture sequence. So what happens here is this total-seq-B antibody capture sequence is complementary to capture sequence 1 on the 10x gel bead. And then this allows you to do simultaneous gene expression with protein expression. In our total-seq-C catalog, we have over 500 catalog products online. These are compatible with the 10x Genomics single cell 5' reagent kits, so V1 and V2 and whatever other version, hopefully, that they come out with. And this enables you to do something called immune profiling. And so in this case, these total-seq-C antibodies are complementary to this template switch oligo, which is present on the 5' gel bead. And then what you get out of this assay is to get gene expression. You get the surface protein expression, a feature barcode, if you will. And then also you can have the sequence of TCR or BCR, depending on which one you would like and which one you'll work with the GEC to do in terms of library prep for you. Okay. So another little example here, looking at enhancing the immunophenotyping via single cell sequencing. So when this sample was the t-SNE was performed using top 100 differentially expressed genes. And so we get clustering of cells. We can see the CD4 and CD8 cells are kind of a little bit intermingled among one another, but that makes sense because they're T cells. So they're going to share a lot of similar gene expression patterns. And then there's clustering of some other cell types, B cells, monocytes, NK cells, PDCs, conventional DCs, basophils, and neutrophils. Now, when we do that again, this t-SNE performed using only the total-seq antibodies and not the gene expression profile. You can see these really neat little tight clusters of these different cell types based on the surface markers that are expressed. And so you can see, your monocytes, different monocyte populations, those B cells, the CD4 and CD8 are really cleanly defined at this point. And then you have our NK cells and little smaller populations of your neutrophils, basophils, and PDCs. Okay, so our single antibody conjugates come in a liquid format. They're in PBS, 7.2 pH. They have some azide and EDTA. And these are tested using flow cytometry and Sanger sequencing. So we know the oligo is what it is by sequencing. We know that the clone binds what it does read out by flow. So we assess that performance compared to our fluor-conjugated versions. These are list price of $369 for 10 micrograms. We know for a fact that you can use less than or equal to 1 microgram per million cells when you're doing labeling. Most people are not labeling many millions of cells for this assay. Definitely contact me for guidance on titrations because we want you to titrate. So, 10 micrograms seems like a small amount, but really it's going to be enough to do plenty of tests. So definitely get in touch with me if you're unsure of how you would go about doing a titration. And so benefits here to these single antibody conjugates. We have the largest single antibody portfolio on the market. They're really easy to use, excellent test value, and you can pool them to make your own panels. And the nice thing about these single antibody conjugates is that, if you have a flow cytometry panel with a bunch of different clones that you buy from BioLegend, you can just look to see if we have the oligo conjugated version of the same thing. You don't need to worry about making a panel in this regard. All you need is a list of markers. All those oligos that we have are compatible with one another. There is no overlap in those barcodes between all the different antibodies in our catalog. So if you want to combine 20, 25, 40, 50, you can put them all together just by picking the list of markers you want and then putting them together in a tube. All right, so now I'll talk a little bit about cell hashing. So what is cell hashing? So working schematic here, cell hashing allows you to multiplex different samples. So if you have 3 different patient samples and you want to combine them into a single capture lane on the 10X instrument, you're able to do that by using these reagents. And so in this case, you'd collect samples from different donors or conditions and label each with a unique TotalSeq hashtag reagent. So in this case, we have, we'll just call them hashtag 1, 2, and 3. And so then we can take those samples and combine them together because these are designed to have coverage of all different cell types. And so we can put them together from the 3 different donors and do a single capture on the 10X instrument. And then we can demultiplex the NGS data to identify the sample of origin. And then you can analyze data from each donor or condition together or individually, up to you. So again, these hashtags target ubiquitous proteins to enable the best cell coverage. So in terms of human, there are 2 clones against anti-CD298 and beta-2 microglobulin, and in mouse it's CD45 and H2 MHC class 1. And so prior to using these, we would always recommend that you try a fluor conjugated version of the clones that are in the hashtag mix so that you can make sure that your cells of interest express the markers that these target. If your cells don't express the markers that the hashtags target, you can't use them, unfortunately. But cell hashing allows you to combine these samples together. So this is sample multiplexing. These conjugates are pre-mixed and ready to use. So in the case of these hashtags, both antibodies have the exact same antibody barcode. So you can't tell the difference between the 2 because we just want to be able to label all cells within the mix. And so as a proof of concept here, cell hashing was recovered expected populations. We had a collaborator, John Ashton at University of Rochester, do some testing for us early on, where he took, he had these populations of bone marrow from different patients. And unfortunately, what he ended up having is that he didn't have a lot of cells for each of the samples. And so because of that, he was not able to basically do a full 10,000 cell capture, and he didn't want to waste, his sample and his different lanes. And so by doing this, he was able to combine it to fill up the space in that lane by combining 2,500 cells basically from each of the samples. And so in this case, he had patient 1 at diagnosis and relapse, and then patient 2 again at diagnosis and relapse, labeled each of these samples with a different hashtag, did sorting for CD34 positives, and then pooling the cells. So he, in this case, pooled 2,500 from each of the samples together and captured 10,000 cells on the 10X instrument. And so in this case, he did also 2 different things where it was stained with hashtags either before or after sorting. And so we note here that there's no loss of the antibody during cell sorting. So I know if you are going to have a small number of cells that are cells of interest after a flow sort, I would always advise labeling with the hashtag prior to it so that the cells are handled as little as possible after they come off the sorter. But anyways, so in here we have a pre-sort HTO stain and a post-sort HTO stain, and you can see that, again, these are very similar. There wasn't a lot of difference between the 2. And the antibodies could still identify the samples. And in this case here, we have patient 1 and patient 2 at diagnosis and relapse having different profiles. And so the ability to be able to do all of this together and then still differentiate from the different patients and at diagnosis and at relapses really enabled them to get a lot more information from what they're trying to glean. And so they're expanding this data set, working on some refractory and remission samples, et cetera. So hashtag advantages. We can identify sample origin. We can multiplex separate samples in the same 10X lane. We can superload the lane so you can actually do more than the 10,000 cells. Helps eliminate batch effects and also helps you to discriminate doublets, because if you have multiple hashtags in the same droplet, you've got more than 1 cell in there. They're pre-mixed, ready to use, and we have them available in all 3 formats for mouse and human, A, B, and C. So last couple notes here, just showing an example of the RNA in this t-SNE plot here, and you have the different hashtags labeled by colors. And you can see here that you can cluster based on the hashtag, so you can separate all those different samples out. And then anything in here where we've got these little tiny dots, that's multiplet, so we can get rid of those from our analysis because we know we've got more than 1 cell in there. So quick note on sample preparation. So we know quality is key when we're doing single cell RNA-seq experiments. Garbage in is going to be garbage out. We know sample viability is very important, and allocating reads to dead cells is a waste of money. So there's a lot of considerations: sample treatment, digestion, cell sorting, sample compatibility with hashtags. Do the clones react to your sample type? Pilot studies and prior optimization can help address all the above. And I am here to help with anything that you would want to talk about. I would rather have many, many conversations with people working on the setup of this experiment than to have to do a lot of troubleshooting at the back end because there was something we could have helped with before the start of the assay. So if you're interested in this, I am always here to talk to, and you can get my contact information, from the presentation and as well from the GEC, they have my info too. So I'm happy to help with whatever I can. All right, so we're going to now talk about how many targets you can look at together. So the multiplexing capacity, because when this first came out, people said, okay, well, that's great. Well, what's the limit then? How many could you possibly put together? So in the initial publication from the New York Genome Center, they used 14 different antibodies. Again, this was their biotinylated ones with the streptavidins and oligos. Around the same time, another group coined the term REAP-seq, which is really about the same type of thing. They used 82 antibodies in their study. And then a little later on, once we had conjugated a whole bunch of clones, we actually said, okay, let's just throw everything we have conjugated in total-seq A against human, 220 antibodies. We put a little white paper together with BioCompare. And so we were able to do that. But I think after that, we walked it back a little in terms of like what really needs to be in a large cocktail. And so at that point, we started to expand our portfolio to have these pre-titrated antibody panels. So this really is nice because it's going to bring a lot of things together that I'm going to talk about. So when we started going down the road of trying to figure out what we would put in a pre-titrated cocktail, we actually used our hashtags to do antibody titrations so that we could determine the amount of antibody to use by sequencing. You can do titrations by sequencing. It is more costly, obviously. We also have guidance for how to do it by flow. So using clones and then staining by flow cytometry to determine how much antibody to use. But in this case, we did 5-point titration series with different clones using our hashtags to run them all together at the same time. So for example, our CD3, this clone was run at the 5 different concentrations. The yellow is the optimal site-seq concentration that was identified, and the optimal flow concentration is 1x. So that's not necessarily 1 microgram, usually a lot less. That's the concentration we identify as optimal, what we say would be a test for certain conjugates. And so we have identified then optimal site-seq concentration for all the different antibodies that go into these cocktails. And then when we were developing the panels, we sort of tried to have a bioinformatic evaluation for optimizing that antibody titration. So when we put together a panel, in this case, we had an imbalance of the NGS data where this particular CD54 at the titration we used, it was going to create an imbalance in the data because it had a really high proportion of the NGS data coming back as reads from CD54. We didn't want that to happen. So our aim is that the percentage of the NGS data should be similar across all of the tags that are included. And assigning the antibody background became a more streamlined process. So we were able to get everything sort of within a range that we were happy with, and then titrate a whole bunch of different antibodies. So I have a table to break down. There's 100+ markers in each of the different universal cocktails for human. And then on same thing for mouse. So just an example of some data that was generated using a universal cocktail. So this paper was published in Cell looking at COVID-19 immune response. And so there was a longitudinal assessment of circulating proteins and 188 surface protein markers. So this was one of our early versions of our cocktail before we made it a catalog product. And so what they did here was they actually used 5', so our total-seq C product, to look at protein markers, transcriptome, and T-cell receptor sequence in PBMCs from these COVID patients. And so what they created was a single-cell atlas, which is really neat, and looking at the immune response to variation in disease severity and outcomes over time. And so, again, here they did actually hashtag staining of the PBMC pools. They had unsorted and then sorted for memory B and T cells. They did the surface protein staining with our total-seq pre-release cocktail in total-seq C format. And then they did their single-cell RNA-seq capture with the 10x genomics 5' with BDJ, and then sequenced and did all of this analysis downstream. And so what they generated here, they had sort of 30 different coarse resolution immune cell subsets. And then their longitudinal analysis, because they have, a number of these cocktails, they were able to classify the innate and adaptive immune cell subsets over time. And then really refined those clusters based on other surface markers for lineage specific cell identification. So you can see more of this paper in Cell was published in 2021. All right, so I mentioned the cocktails, I mentioned the table, here's the table. So again, in our human universal cocktails, so this is sort of our everything panels, our total-seq A has 154 antibodies, total-seq B has 134, and total-seq C has 130. In our mouse, we didn't want to leave the mice out, so we have 119 primary antibodies in total-seq A format, 102 primary antibodies in B, and then 113 in total-seq C. Hold on, I might sneeze. Okay, no, we're good. And then we have only available for human, but we have these TBNK cocktails, which contain 9 primary antibodies to some sort of major lineages, T, B, and NK. And so those can be nice if you know you're going to include those ones. You can have those as a pre-titrated lyophilized cocktail that you could then add individual antibodies to. So these cocktails are provided in single-use tubes. They come in a pouch. There's 5 tests in a pouch. Each test is in a single vial in a lyophilized pellet. You reconstitute it prior to use. So if you only need to use one, you can keep the rest in the fridge and they're good for like a year, as long as they're stored correctly. Again, pre-titrated and validated using NGS as a readout, validated on PBMCs for human, splenocytes and bone marrow for mouse. And this helps to reduce inconsistency with pipetting and handling multiple vials and samples. It has a reduced cost, a reduced cost compared to buying individual antibodies. And it helps to minimize variability between experiments, between different labs and during longitudinal studies. All right, so key takeaways of the panels. They're convenient. Optimal concentration for staining is already identified. Reduced cost, they're lyophilized for stability, minimize the variability. Then things I didn't point at before were that you can combine them with our hashtags if you want to do sample multiplexing. So as I showed in the example from the COVID study, you can label with hashtags and then do the cocktail label. You can cocktail label, if you label multiple samples and then pool a certain amount of samples, you can use one total-seq cocktail. There's a bunch of different ways that you could do that. You can also spike in additional antibodies. So if you see, okay, yeah, I like the total-seq B mouse universal cocktail. However, it's missing like this one marker that I want to use. How do I go around that? So you buy the universal cocktail, then you buy the liquid antibody, and I can provide you guidance on how to add that in your lab. It's not done by us, but it'll be done by you. I'm gonna, I think, skip over the total-seq D products. If you're interested in single-site DNA-seq, contact me. We're gonna skip over that. Okay. And then I'm gonna go over, since we do have time, the multiomics analysis software, since I did tell you that it was an end-to-end solution. So a lot of people kind of shy away from doing any single-cell RNA-seq because it could seem overwhelming with a lot of the data analysis on the after you get through the whole sample processing, and then what do you do with the data? And so we actually have a web-based free cloud-based web, I don't know, whatever. It's on the internet, it's free. It's called Multiomic analysis software, very creative. So this is, there you go, a free cloud-based program for analysis of site-seq data, so protein and RNA. So you have some data that comes out of 10x Genomics Cell Ranger, a feature barcode analysis workflow, or you can get 10x data that's processed using Seurat. And so basically this just kind of comes down to, this would be all the stuff we talked about beforehand, get the sequencing of the libraries, and then you process and align the data using Cell Ranger. And then with some files that you get coming out of here, then you can use MAS to kind of do some further analysis. So what are the benefits of MAS? This has a simple user interface for exploring the site-seq data. It's accessible to all users, irrespective of your bioinformatics background. You can use your web browser. There's no server access required. You can use flow cytometry-like gating based on the total-seq antibody staining. Create UMAP, TISNE, TriMAP dimensionality reduction plots. You can easily share any workspace with collaborators and our technical service, which is really nice. And then you can export the project in this star H5AD format for use in other single-cell analysis programs. So how do we do it? We have these input files, so feature barcode matrices or Seurat files. And so you have your features file, which contains the description of the features, the ADTs, the cell hashing antibodies, and gene expression ensemble IDs. Then you have your barcodes file, which contains all the barcodes that are identified during sequencing. So that contains cell barcodes. And then you have this matrix file, which contains the feature counts for the cells. So this tells you how many of each of those UMIs were there. And so then these feature barcode matrices are outputs that you get from that Cell Ranger software. So how does this work in terms of a workflow? So we have our data upload. So we go through here, you can add new file set. It asks you to make sure that all these things are loaded. So if anything is missing, it will prompt you. You won't be able to go to the next step. Then you can demultiplex the data using cell hashing. This is the only software that incorporates the demultiplexing of data based on cell hashing antibodies or our cell hashing antibodies. It's a big point that's important here. So it also makes available 2 demultiplexing algorithms, purity doublets derived from the original NYGC cell hashing paper and demux EM. So users can choose whichever one you want. In this case, it's denoted as purity doublets, so you can choose that in the software. And then this will automatically filter and make your filtering more stringent. Okay, and so then you can see here where it's doing this demultiplexing run. And then we have some debris algorithms. So the data sets that don't contain hashing data are automatically filtered via UMI count distribution knee plot. So that's what this looks like. Thresholds used for this knee plot filtering are adjustable by the user. And so we can remove cells from further analysis if they have too many UMIs, so over on the left-hand side, or relatively low number of reads, so too far down on the right-hand side. And that MAS will do this automatically. If you don't have any hashing, it's going to run that. So then we do highly variable gene and high expression gene RNA analysis. So we'll do this filtering and normalization here. So this process helps to narrow down the list of genes that can yield biological insight. And it's done by binning genes as either included or excluded. So excluded genes are masked from subsequent analysis and included genes are subsequently normalized. And so you can see here what ends up getting included. You can change your threshold for the gene UMI. You can change the threshold for the high variability genes. You can search for the genes here. So, if you're going to go ahead and your favorite gene of interest, you really want to make sure that gets in there. You can search for it and see whether it's going to be maybe excluded based on the fact that it wouldn't have high variability or high expression. But you can see at least that it might have been reported. Okay, so then we are going to do some gating to clean up the data and define cell populations. So again, as I mentioned, you can do flow cytometry like gating on the single cell data. So you use these log transformed ADT counts so that you can display them. And so in this case here, we have, I don't know what that is, CD30, CD4? I can't see, I can't tell. Anyways, we can put 2 different, it is CD3. Okay, CD3 and CD19. So we know that anything that's double positive for those 2 is probably not really a cell, or it's dead or something like that, because those 2 should not be co-expressed because they are T and B lineage markers that are mutually exclusive. So we can gate that anything that doesn't have a double expression of those would be probably a real cell. And then from there, you can go ahead and do some gating on specific cell populations if you wish. You don't have to do this part, but you definitely can if you know you're kind of going into this looking for specific cell groups that it would express markers. Then we can also do some cleanup based on isotype controls that allow you to exclude cells that would express multiple isotype controls. Those would be dead or dying cells or things that we don't think are actually real. And then again, further stratify different populations of, for example, T cells based on expression of different surface markers. So in this case, I'll talk a little bit again about those isotype controls. So in the top panel here, we have isotype positive cells were shown in the red circles. So they're there. And these CD3 positive CD19 cells, which I said were probably not real, in this case, were not removed. And in this case, they were removed. And so the isotype positive cells have decreased by removing those double positive CD3 and CD19 things that were probably not actually real in the first place. And so it's more emphasized when you get further out in these plots here, showing there's, a high number of isotype control binding cells, and those kind of are almost, I know it's a really tiny circle, but they're pretty much gone from the circle on the bottom plot there. All right, so then we can go ahead and do some dimensionality reduction with our data. We can quickly and easily create dimensionality reduction plots, performing clustering using community detection algorithms. The determined clusters are projected into these plots. And MAS, like I said, can create UMAP, TISNE, and TriMAP plots. So the cells are plotted in a feature space. The features are things like ADTs, hashtag oligos, or genes. The dimensionality reduction results in those plots, and clustering is the grouping of those cells based on similarity of dimensionality reduced data. All right, so then users can view the expression of both RNA and protein data, and they have the expression data overlaid on top of the dimensionality reduction plot and clustering results, so you can easily identify different cell types. So you can have the UMAP clusters here, the protein data, the RNA data, you can combine them together. You can inquire where you actually like go and draw a little circle around a particular component, and then you can go ahead and look at the expression, differential expression between genes. So if you take, different clusters, you can have, a plot like this where you would compare the difference in gene expression between, cluster 1 and cluster 2. You can see orange is protein and blue is gene. And so you can see where there's significant differences in both or either of them. And then you, like I said, can compare 2 or more clusters to identify proteins and genes that distinguish them. You can have fold induction reports and cluster in the top window or the bottom window. All right, some questions that we get sometimes on the MAS. We don't have the ability to analyze multiple data sets at the same time. We're working on enabling merging the data set functionality, but we can't put them all together right now. It can also not handle aggregated data files. Well, it might be able to, as long as the feature file is following the same rules and conventions in the manual. We cannot analyze data sets that don't have total seq data. So if you have just single cell gene expression data, no total seq or site seq involved, we don't have the ability to do the analysis because it is looking for that file that contains the barcodes. And then we also have an example data set. So if you're interested in playing around with this, you can go and log in and make a user profile. I can send you the data set that we have, which is human PBMCs labeled with our human universal cocktail and total seq format. And then you can kind of fiddle around with it and play with it and see how the software can perform and what you can do with it. All right, so I think I'll spend a couple more minutes doing one more thing here, and then I can leave it for questions. So the last thing I'm going to mention is our dead cell removal kits. So as I mentioned before, high viability and good sample viability is important for single cell RNA-seq experiments. So we have these kits that allow benchtop magnetic removal of dead cells prior to doing site-seq. So if anyone's familiar with these from doing cell enrichments, we have these MojoSort kits. They have these little magnet. It's basically you have cell labeling and then you put them in the magnet and you can pour off cells. And so our dead cell removal kit has these monomers and streptavidin nanobeads that we make a complex with before you incubate with the cells. You do this in our standard MojoSort, which is very similar to cell staining buffer. So it's like PBS with some serum. You don't need any calcium added like you would use for anything that's annexin 5 based. This is not annexin 5 based. And so in this case, you do this incubation, you do a wash, then you take that tube, you put it into the magnet, and then after an incubation, you pour off the live cells into a tube and all the dead cells will be remaining inside the magnet. So in this plot here, we're showing mouse dead cell removal kit and human dead cell removal kit. Pre and post enrichment. So before enrichment, we had 64% viability, and after enrichment, we have about 97% viability. We're really reducing the positive markers for cells for the Helix Blue dead cell marker here. And this is just comparing Helix Blue and Apotracker Green to identify apoptotic cells. And so you can see that there's a lot fewer of them after the enrichment. And then the human kit, same thing. We really increased the proportion of live cells quite significantly. And this is without doing flow cytometry. So our kit versus some competitor kits. So we can see here our live cell yield is higher than a competitor, and for both the human and the mouse dead cell removal kit. And this is just showing how we've improved single cell RNA-seq data and site-seq data. And so we have 3 different sample types here. Fresh would be fresh live PBMCs from a healthy donor. Then we have a live 60 group, which is PBMCs at 60% viability, where we basically heat killed cells and we combine them with fresh PBMCs at a 60-40 ratio. And then the dead removed, where we took the previous sample and then used the Mojasort human dead cell removal kit to remove those dead cells that we had added back in prior to doing the experiment. And so the proportion of higher, like, so a high percentage of mitochondrial gene reads relative to total gene reads, if it's higher, it means there's more dead cells. So in this case here, we have our fresh sample where there's not as many mitochondrial reads. The live 60 group has quite a few more, and the dead removed looks back almost like the fresh one. So showing a UMAP plot where you can see clustering of different cell populations, we can see that the dead cells all cluster together, and those are in the live 60, not in the fresh and dead removed samples, which is hard to see, but green is fresh, orange is dead removed, and blue is the live 60%. So there's all those dead cells there. And again, when you cluster those together, you can see mitochondrial gene reads are lighting up in that dead removed cluster. So another thing that this improves here is the binding of different antibodies for site-seq. So again, we have our fresh in green, live 60 in blue, and dead removed in red. And so this is just showing binding of different antibodies on the dead cells. So you can see that there's a difference in the expression of these different markers when we have a lot of dead cells involved. And so this really will allow you to clean up your data by removing those dead cells prior to your experiment. Okay, so with that, I'm going to open it up to questions. I'll try to go back to my slide that has my contact info. And then I guess, yeah, Sandra can read some questions. And if there's any in the room, I'll take them as well. Thank you. Mm-hmm. Yep. So our magnet only holds one, but if we discuss, we can talk about potentially using other magnets from other companies.
There are no questions in the chat.
Sure.
Is there any specific guidance for people who are interested in doing site-seq with the new fixed cell?
Not yet, but we're working. Not yet, but we're working on it. I would suspect. Okay, how am I going to say this so that I don't want to get in trouble? So on the fixed RNA kits, there is a fixed feature barcode kit you can buy from 10x, correct? So they have some demonstrated protocol using total-seq B antibodies. So the guidance would be basically what their protocol is. The cells have to be labeled before they're fixed. So if you have fixed samples that have been hanging around for X amount of time, you cannot combine the antibodies. We don't know whether all the clones or antigens will be preserved after fixation. So like we don't, or if the antibodies will even bind. So we advise staining before fixation. Now I have had questions asked whether or how long you can have the cells stained, fixed, and then store before you actually do your acquisition. And that I don't know the answer to, and I don't know that 10x knows the answer to that either. So something that we can definitely discuss. You know, it's something that we have in our teams that are trying to work on it, but we don't have an answer just yet for that.
Okay, well, thank you very much, Leesa. We
welcome.
appreciate you coming today for the Biotech Talk this April. And we look forward to hearing more from you as our clients reach out to us to use the BioLegend products for their single cell RNA experiments. So once again, thank you. And I'd like to remind our people attending this meeting that next month's Biotech Talk is now on May 16th. It's going to be a two-part talk. The first talk starts at 10 a.m., followed by 11:15. The 10 o'clock talk will be given by Holly Center from Thermo Fisher Scientific, and her topic will be digital PCR fundamentals, how digital PCR can complement your qPCR experiments. And followed that will be a talk given by Dustin Rubenstein, the director of the UWBC Genome Editing Center. And his talk is titled Using dPCR to Confirm Genome Edited Models. So once again, that will be on May 16th. And also on May 4th, there will be a 10x webinar that is being co-sponsored between 10x Genomics and the Biotechnology Center. That will include us talk a brief overview of the services available in the Gene Expression Center for 10X applications, followed by Egon Roney from 10X, who is one of their application specialists, and which will also then be followed by two speakers of our 10X genomics, 10X genomics applications. One of those speakers is Kristen Gantz. She's a PhD postdoctoral fellow here at the University of Wisconsin. And then another one by Irene Ong student. So once again, this will be on May 4th here as a webinar. So thank you again, all of you, for joining us today, and I hope you have a nice day. Bye-bye.